Hello again, this is Dr. Dobbs McAuliffe. We're continuing on with Chapter 12, looking at transport across cell membranes. So I've posed the question here, what allows a molecule to cross the phospholipid bilayer? This would be things that can fit between the phospholipids. So if I were to draw a phospholipid bilayer, phospholipids are hard to draw with a trackpad, but hopefully you get the idea. This is a phospholipid bilayer, and that there are spaces between the phospholipids. Some things are able to pass between the phospholipids according to their concentration gradient. So some things can sneak between these phospholipids, and it's going to depend upon some basic characteristics. So we can see that small, nonpolar molecules, things like oxygen and carbon dioxide, steroid hormones, and other gases, can easily pass in between the phospholipid bilayer. And then small uncharged but polar molecules also can pass through water, ethanol, glycerol. But if you notice, the arrow is not as thick, the blue arrow as compared to the green. This means that it's much harder for water, for example, to cross the phospholipid bilayer. It's not going to move as quickly or as many at a time. Larger, uncharged polar molecules, things like amino acids, glucose, and nucleosides, cannot really readily pass the phospholipid bilayer. These aren't extremely large things, they're just too large to fit between the phospholipids. Ions are small, but the charge and their extremely hydrophilic nature means they cannot cross the phospholipid bilayer. One way we can kind of characterize it is for each ion or molecule, whether or not it can pass the phospholipid bilayer is determined by its size. Is it small enough to fit between the phospholipids? And charge. If it has a charge, no matter what size it is, it's not really going to readily cross. Now I have some notes here on size because I want you to realize that even the things that are saying are large are not necessarily large when we compare to other things within the cell, like proteins. So 1 Dalton is about the size of 1 proton or 1 neutron. Oxygen gas is 2 oxygen molecules bonded together is 32 Daltons. Water and oxygen and 2 hydrogens is 18 Daltons. Oxygen and water can both cross the phospholipid bilayer. They're small enough. Glucose is 180 Daltons. It's already too large to cross the phospholipid bilayer by slipping between the phospholipids. Typical proteins range from about 1,500 to 250,000 Daltons. And I found this number by looking for common molecular weight markers. But some can be much larger, close to 4 million Daltons. Only atoms and molecules of a small size are able to pass through the phospholipid bilayer. Glucose isn't necessarily a very large molecule, but it's too big to fit across the phospholipid bilayer. Proteins don't pass through the phospholipid bilayer. They're too big. Here I've come up with a few hypothetical examples of how a molecule could cross a phospholipid bilayer. So I'm giving you an example in this picture and saying that blue is a small, nonpolar, uncharged molecule. This would be something like oxygen or carbon dioxide. Imagine this is maybe a red blood cell. If we were in a high oxygen environment, so if there was a high concentration of oxygen outside the cell, there would be net diffusion across the phospholipid bilayer into the cell. Right? So blue molecules would be moving in. Now if we look at the red molecule, it is a large polar molecule. The concentration gradient would allow it to move out of the cell, but it cannot cross the phospholipid bilayer. Gray, I'm saying, is an ion. The gray molecules, like the blue, would move from outside of the cell to inside of the cell, but they cannot cross the phospholipid bilayer. For each of these solutes, they would tend to move down their concentration gradient, but only the blue, in my hypothetical situation, could actually cross the phospholipid bilayer. So what does this mean for the cell? So only some molecules are diffusing down their concentration gradient. And so the only way to control where those molecules are is to control the gradients themselves. So this is what's happening in our lungs. We breathe in air with a high partial pressure of oxygen, and the oxygen can then diffuse down its gradient from in the interior of our lungs into our red blood cells, into the plasma, and then be brought to other parts of our body. However, for ions and larger molecules, those can be contained within our cells, and there's more control as to where they are located. We can control the release, the flow of those ions across the membrane down their concentration gradient. We can also pump them against their gradient and kind of store them up in particular regions of the cell. So this is what actually does happen, and we have really large differences in intracellular and extracellular concentrations of particular ions. Inside the cell tends to have a high potassium and hydrogen ion concentration, whereas outside of the cell is high in things like sodium, magnesium, calcium, and chloride. You may note that the concentrations of sodium and potassium, they kind of mirror each other, right? So sodium has a concentration of 145 millimolars extracellularly, and potassium 140 intracellularly. Sodium has a low intracellular concentration of about 5 to 15 millimolar, and potassium is about 5 millimolar in the extracellular fluid. So these concentration gradients are similar magnitude. The difference in concentration is similar, but the direction is opposite. I have a note here. Overall, though, if we're looking at a cell, the concentrations of positive and negatively charged molecules in the cell are balanced, except for local differences at the cell membrane, which is going to become important in action potentials. So these gradients that I just showed you, that, for example, the large differences in the amount of sodium in the cell or potassium in the cell, there's large differences in calcium, which is much more abundant outside of the cell than inside the cell. Muscle contraction relies upon a release of calcium. Action potentials, so that is the electrical signal that travels down our neurons, and the production of ATP. I believe Dr. Smith is going to go over that in detail, looking at what's happening within the mitochondria in particular, but through the process of glycolysis in the cytoplasm, citric acid or Krebs cycle, where those products move into the mitochondria, and then electron transport chain and oxidative phosphorylation. Oxidative phosphorylation requires a hydrogen ion gradient. You may have also learned about that in other classes. An ion can't fit between the phospholipids. Neither can glucose, for example, but they do get into our cells and they do get out of our cells. So how do they do that? Well, there are two classes of molecules that are involved. They're both proteins. So this is a channel protein. So the protein is a channel, and it can allow a space for ions to cross the phospholipid bilayer. So they're transmembrane proteins with a pore or space in the center. They can be open or closed, sometimes called gated. Transporters are also proteins, and they have a binding site for a particular solute in the cytoplasm or extracellular fluid that's going to be transported. They are not open all the way through. So they switch between two different shapes. In this case, the transporter is open to the intracellular fluid. It can change shape and then be open to the extracellular fluid. So they're only open to one side of the bilayer at a time. Another aspect of transport that is important to think about is whether the transport is active or passive. And this is really determined by whether the molecule is being moved with or against its concentration gradient. So the first thing we looked at in this chapter was simple diffusion. Things like oxygen and carbon dioxide and water can move in between the phospholipids. And that's always passive, and that's based on a concentration gradient, right? So the green is going from high to low. This yellow molecule is going to also be transported across its concentration gradient, or down its concentration gradient across the membrane from high to low, and it's going through a channel. This is passive transport also because we're going high to low. The channel is just creating this space, a hydrophilic space for that molecule to move through, for that ion to move through generally. A transporter is also creating a space. It's through a binding interaction and then a change in shape, but it's also passive transport. So simple diffusion and channel-mediated and transporter-mediated are all passive transport, but the channel-mediated and transporter-mediated would be considered to be facilitated diffusion. There's still diffusion, which means it's passive, but you need kind of a helper. You need to create something like a doorway. So a channel I think of as like doorways, which can sometimes have a door or a gate. I think of transporters more like revolving doors where they're never open to both sides, the inside and outside at the same time. Active transport is characterized by moving against the concentration gradient. This blue square is at a low concentration inside the cell and a high concentration outside the cell, but we're still going to keep moving it in that direction. So to do this, there has to be an additional source of energy to pump. So think of pump as something you have to plug in, right? That's something where you need additional source of energy, and this is going to allow for movement from low to high concentration. For simple diffusion, channel-mediated, facilitated passive transport, and transporter-mediated, facilitated passive transport. In all of these cases, there is energy within the gradient itself. So what type of things can be moved across the cell membrane? Here are some examples of things that are moved through channels and transporters in a cell. So we can see nucleotides, sugars, amino acids, ions, sugars. We can move within the mitochondrion, within the lysosome. So there is transport across the cell membrane, but also across the membranes of organelles. So one of the transporters we're going to look at in more detail is a transporter used to move glucose across the phospholipid bilayer. Remember, glucose is too big to cross in between the phospholipids, so it needs help. It's facilitated by a protein transporter. Now in this picture, glucose is the blue. And glucose is going to move from a high concentration to a low concentration. So this is facilitated diffusion, no added energy. This particular protein is highly specific to binding glucose. It's not going to transport other molecules. So here glucose can enter the binding site, and then this protein can randomly switch conformations. It'll go through an intermediate where it's closed on both sides, and then in this case open to the intracellular space. The protein will then switch shape again, but this time with no glucose in the binding site, and then return to its original conformation. So this protein is highly specific for transporting glucose. It can switch randomly between 3 conformations. We will get a net movement of glucose from high to low concentration because we're only likely to have glucose binding on the side where there's a high concentration. This allows glucose to move, in this case, from extracellular to intracellular space. If the gradient was reversed and we had a high concentration of glucose within the cytosol, it would then work in reverse because we would have a very high likelihood of glucose binding from the interior of the cell, and then the transporter can move it out. So this is passive transport, and it's always going to move glucose down its concentration gradient from high to low concentration. We will see this transporter in use in transporting glucose across the cells of our intestine, so from our food through our intestine to the blood.